Today we are going to see the topic disorders of the excretory system. Okay, that is kidneys and nephrons. The topic starts, that is malfunctioning of kidneys, means the kidney is unable to function properly. Then what happens? Accumulation of urea in blood rather than going outside in the urine. Urea is settling in the blood. That is a problem. Okay, this has to be concerned. So this condition is called as uremia. It's called as uremia. Topic starts. That is hemodialysis. We'll see soon. That is disorders of the excretory system. So what is the first point? That is uremia. What they are saying uremia? That is urea will accumulate in blood. This is important. In the blood. Okay. Next, this is harmful and lead to the kidney failure also if it is not absorbed and not monitored and not cured. In such patients, urea can be removed. The process. Now, what is happening? That is, urea can be removed artificially by a process called as hemodialysis. Okay, that is blood diluting and removing. So this process of hemodialysis, in that what's happening means they remove the blood from artery. Okay, and they'll send to the one unit, a machine that is called as dialyzing unit. Okay, then the blood is removed now from artery. In that they'll add a heparin. What heparin will do? Heparin will not allow the blood to become coagulant. Okay, it will make it to liquefied only. So then the blood is sent to the tube, cellophane tube, like a reverse osmosis. Okay, it has a pores. Okay, around the dialyzing fluid. I'll explain now. You see, this is a machine. Okay, first is this is the method. They'll take from the artery. Okay, like this. They take it from the blood artery and they'll add a heparin so that the blood will be in the liquid form only. It will not become solidification. Then it is pumped. It's a motor. Pumped to the a tube. These are tubes are there. See, a tube will be there. So the tube will have small holes, minute holes. Okay, and here outside there will be a fluid will be inlet and outlet. So what's happening in this? When the blood is moving through the whole pores of these tubes, then it will come out. So while before this, what happening means, because in the dialyzing fluid it will be equal to the blood, plasma, proteins, cells, everything will be there except nitrogen. So here in this, the nitrogen will move. If you want to say how it will mean, this unit I am drawing. So it will be like this. Suppose this is the unit. In this tube will be there like this. Okay, the blood is pumped from here and the blood is taken out, suppose if you think. Then what happens? Here in this, the outside is liquid, this open space has no nitrogen. So nitrogen will move from this tube to outside. So while this, the blood will become free. So what the blood is going, it is from this, the nitrogen is removed, it is going out because of diffusion. Then the blood is filtered. So this is the case. See here, what is happening? They're taking the blood, okay, and they're sending into this fluid here. In this machine, this is the liquid is coming up. So here somewhere it is connected here. From here it is going. So what happens when the blood is moving from the tube? Nitrogen will go outside. Only nitrogen, because remaining everything is same. Then once nitrogen is moved, then it is free of nitrogen. Then the blood is sent back again. Okay, it will move because moving is like a pumping heart. Like it moves, it rotates. So liquid will be moved outside or inside. Then the blood is again taken and given to the vein. So this is the hemodialysis. Okay, see, the blood is taken from artery. Okay, pulsar is checked and heparin is added. Then here in this liquid it is rotated. So while rotating and coming out, while moving, nitrogen will come out into the outside liquid. So that will be fresh. The fresh liquid will be sent here, and bad liquid will be sent out. Okay, like pouring the water in the aquarium like this. So while moving, the nitrogen will be taken into the outside liquid, and the liquid is drained out, and new liquid is sent. Then after that, that blood shows the blue color means it's a free of impurities like nitrogen. Then again sent. Before sending, they'll add anti-heparin. Here they'll add actually. Here heparin added, anti-heparin. If they won't add anti-heparin, the blood will not be liquid form much. It will not coagulate. Further, they have to add anti-heparin. Then they'll send back to the vein. This is the mechanism. So here's the same mechanism. Taken, okay, sent inside, and it is sent here. It's filtered here, and it is sent back. This is the mechanism. This is an example of the dialysis unit. Okay, so this is the process. So now I will make notes for you. Okay, so what is saying it is, first we'll start. 1 second. The topic is what? It's a hemodialysis. Hemo dialysis. How it happening? How it is happening means first the dead artery. You see, okay, the composition of the plasma except nitrogen waste, the porous the membrane that tube will have a holes. Okay, because of this porous cellophane membrane of the tube allows the pass of molecules based on the constant gradient, means based on the amount. Suppose example, again I will draw here. This is the dialysis chamber. Okay, in this the tube is like this. Okay, the tube will make the tube. Like this. So here nitrogen will not be there. In this nitrogen will be there. So when the nitrogen is here like this in the tube, it is jumping outside. So nitrogen from the tube, it is jumping. So nitrogen will move outside. So here it's free of nitrogen. So this is the artery. Then this is a vein. So blood is sent, taken out. Okay, and this fluid will be drained out. This is method. So this is what is saying that the molecule, because it's here what is happening, nitrogen is high in the our tube. Here nitrogen is less. So because of diffusion, high concentration, low concentration, the blood is from the blood nitrogen is moving out easily. Remain everything will be similar. Here also plasma in this will be equal to the plasma in the dialysis unit. So there is no change. So only nitrogen will be less here. So a nitrogen waste are absent in the dialysis fluid here. The substances freely move out passively, thereby clearing the blood. The cleared blood is pumped back to the body through a vein, adding anti-heparin. So it's a very boon for thousands of uremic patients all over the world. Even some states in India, they are giving subsidy for the patients so they can go for this treatment freely. Now let's make notes for this. Okay, hemodialysis, very simple. One is artery. First from artery they'll take the blood, so that's important. Then the blood is taken, any artery from the hand or leg taken out. Then heparin will be added. Heparin is added. It is what actually, it's an anti-coagulant. Next, it is sent to the dialysis unit. What is this? It is a artificial kidney. It can be also called as artificial kidney. Next, fourth. What is in the two things will be there? One is cellophane tube. Okay. There's a two things. That is cellophane tube. Fifth, it has dialysing fluid. Dialysing fluid. So what is cellophane tube specialty? It has a porous in nature. It is porous. It has a holes. Dialysing fluid except nitrogen. It is except nitrogen equal to the blood. So this is enough. Try to remember. Next, what happened? Number 6. Okay, that is here happened. Process diffusion occurs. Okay, it means it removes. Okay, nitrogen. Next is what? Nitrogen will be sent out. Nitrogen moves outside. Into where? Into dialysing fluid from the cellophane tube. Next, 8. That is blood is free of nitrogen. Okay. Free of nitrogen. 9th, what they'll do, last step, anti-heparin is added. Okay, anti-heparin is added. It's important. 10th, into the vein. Okay, returns. Okay, blood is returned. So this is the 10 steps. So remember, from artery the blood is taken out, added heparin into dialysis unit. It has a cellophane tube, dialysis fluid. So because of diffusion, nitrogen will be moving out into dialysis fluid. Then the blood is free of nitrogen. Then anti-heparin added, and the vein into the blood is returned to the vein. Simple. So these 10 steps you have to remember. That's very easy. Hope you understood. This is just high concentration to low concentration. Simple. Next is the kidney transplant is the ultimate method in the correction of acute renal failures. That is in the kidney failures. Okay, if suddenly all of a sudden, okay, the kidneys failed due to many reasons, any reason, so they have to replace the kidney. But a functioning kidney is used in the transplantation from a donor. Means a good kidney. Okay, preferably a close relative only, not from outsiders, because our body will understand the cells, that is our cells, okay, other cells, that is like everything whatever enters the body, our body will assume it as like antigen, okay, outside material. So that's why they will not accept. So to minimize its chance of rejection by the immune system of the host means the person who will receive the kidney. So if we unknown stranger kidney, it will not accept. So only close relative kidney will be accepted. That too we have to check. Okay, the doctors will check with the method whether the body will accept or not. If it is accepted, then it's good. If it's not accepted, then also they will replace the kidney, but with the help of some immunosuppressing agents. So to minimize this, the modern clinical process have increased the success rate of such complicated technique. Okay, nowadays that's possible with some drugs. Next, example, renal calculi. Renal, it's a kidney. Calculi means calcium materials. That is stones, calcium stones are insoluble, okay, which are unable to solubilize. Mass of crystal salts, oxalates found within the kidney in the medulla. Okay. Next, this is another. Second is what? That is uranium. Okay, it's a uranium. What is saying? It is already uranium completed. So again, we'll come back. Second is what? Renal calculi. like what it's saying, it contains stones or crystal salts like oxalates. This you remember, okay? You can write 1, 2, 3 also, but much better. Stones or crystal salts like oxalates will be there in the kidney, renal ganglion. Third, okay, that is glomerulonephritis. That is the glomerulus in the nephron, okay, has some problem. What is the problem? Inflammation of glomerulus. Many glomerulus, malfunctioning bodies. That's a problem in the kidney. So that is the answer is inflammation, swelling, okay, of glomeruli. Many glomerulus. Okay, this is the case. Not only this, even the last page, these are the disorders actually. So even it's better to remember, that is the fourth one, glycosuria also. Okay, fifth one is ketonuria also, we should remember. Why? Because in the last paragraph they mentioned that is because of problem in the kidney functioning. Glycosuria. Okay, what is saying? That is glucose in urea, in urine. Next is ketone bodies. Second is ketone bodies, okay, ketone ketones in urine. This is difference. So uremia and glycosuria and ketonuria. These three things you have to remember. If you want, you can keep this also here. It's much better. Or stakes if you don't wish. Okay, this is the concept. I hope you understood this topic. Okay, very simple concept. And this chapter completes. Okay, thank you. Have a nice day.